Dr. Alan Wood joined BTI in 1968 after receiving his PhD from Purdue University. His early research included the first documentation of the divided genomes of component plant viruses and studies on double-stranded RNA viruses of plants and fungi. His work in insect virology led to the first U.S. field release of recombinant virus in 1989, using a recombinant baculovirus to control cabbage loopers. Allen co-founded Agravirion Incorporated in 1993, a biotech company producing pharmaceutical proteins and larvae. He later became the founding director of the Life Sciences and Biotechnology Institute at Mississippi State University and participated in a USDA task force recommending the formation of a National Institute for Food and Agriculture.
I did an undergraduate research project at Middlebury College where I was I was looking at a nematode-trapping fungi that David Pramer at Rutgers set me up to do. Turns out he's a pretty famous guy. I don't know why he took all his time to spend time with me, but anyway, I did this project, and I realized I hated looking in a microscope. My stomach would get kind of wheezy. So I knew when I went to graduate school that it was not going to be in a subject that had to do with anything to do with a microscope. So I was offered a position in a plant virus lab at Purdue. I was like, perfect, don't have to look at any microscopes at all. So that's kind of how I got into it, kind of backwards. One of my colleagues at Purdue was offered a job at the Boyce Thompson. Harry Mussel was offered a job with Dick Staples. He came back and he told me, he said, well, maybe they've got other jobs open. So I sent a CV along with my all the information that they could possibly want. Never heard a word. Then all of a sudden I got an invitation to come for an interview. They blew me away. When we went out to dinner, every faculty member was at the dinner. We went to lunch, every faculty member was there. It was a mausoleum full of really modern equipment. It was really great. And they had a project that they were interested in me working with somebody else to work on. So my exit interview with George McNew, he essentially said, we'd like you to come. Would you be interested in working on that project, if you came here? And I said, no. This is my first and only interview I ever had, but I knew I did not want to work on that project. So George said, well, what would you work on if you came here? So I told him that I was interested in doing this genetic study with plant viruses, and we had done the crosses. Well, I wanted to do, but in order for a true genetic study, you need to do a back cross. And these are big projects. The first one was big, the second one is even bigger. And so he said, oh, that sounds good. Why don't you come and do that? I mean, what more could you ask for? Somebody's hiring you to do the work you want to do. That's what I did for 34 years. I only did what I wanted to do. They let me, I got bored with something or not totally uninterested about something after a while, and I was changing the subject. They didn't care. Of course, I had to bring in money and had to publish papers. That was the important thing. But I had like 4 different careers at Boyce Thompson. When I first started out, I was doing the recombinant work with the genetics of the plant virus. We were finishing that up. Actually, George McNew came. He come from a meeting in England, and he was interested in maybe the fact that plant fungi that are pathogens might have viruses in them. It was an interesting idea. So he said, here's some money, and investigate if there are any viruses in fungi. Well, there are. Bob Bozarth, who I shared a lab with, he and I joined 3 other people in the world. The other 3 were in Europe, the only people that worked on fungal viruses. And that's where we got into double-stranded RNA. They were all double-stranded RNA viruses. It was people at that time were very interested in what viruses were in fungi. It was like a trip around the world. And it was ridiculous. I got to see a lot of the world because of it. But then I got to the point where I got bored. because I couldn't transmit the virus to another fungus to see what the biological effect was. All I knew is, well, the virus is there. Is it doing anything? I don't know. And so there's no biology. It's all physical chemistry and stuff. I just got bored. So that's how I went into insect viruses. There were 2 other people working on insect viruses at the time here at the Institute. Neither one of them were trained in virology. So this would be perfect, here, add a virologist to the group. I took about a month just to study insect viruses in the library and decided that there's some really interesting things to do. They would never, ever be used as a way to control insect pests in the field. I thought it was ridiculous. Well, it turned out, I decided it wasn't that ridiculous. I ended up doing that, precisely the same thing. Genetic engineering was just coming into vogue at the time. And the idea became, insect viruses, when they infect an insect, they kill them very slowly so that the insect continues to do damage to the plant. By the time it dies, it's too late. Most of the damage is done. So the idea is you need to speed up the killing of the insect or the pest. The way you do that is insert a new gene, like a scorpion toxin, into the insect virus. So when it infects the insect, the scorpion toxin is expressed, kills the insect very quickly. In my mind, the problem with that is what happens ecologically. There could be a problem. And so I started trying to think of ways in which we could investigate these things and look at the ecology of these viruses and the insects in nature, the possibility that we could develop something really useful. So that is when I approached the Environmental Protection Agency, and they gave me a grant to do a field release I had this idea, this theory of how you could have an engineered virus that would not cause any environmental problems. We needed permission not only from EPA, everybody else in EPA, USDA, everybody and his brother. I've got a book about that thick of permissions. Nobody raised, it wasn't really much of a problem. But then the press got a hold of this. The Cornell Press were amazing. They sent over copies of hundreds of articles written in newspapers and magazines around the world about us going to release a recombinant virus into the environment. It blew my mind, it really. Actually, Collier's Encyclopedia had a whole article about it. My father-in-law gets the Collier's Encyclopedia. When he read that, he was pretty impressed. He said, "Oh, my daughter married a really important guy." It was kind of funny. And then we had, dozens of people come for television stations for clips. Martha Hamlin was working with me at that point. She did the overflow. I was always on the phone at all, and she would answer. She was fantastic. She actually went on and got a PhD in genetics at Cornell after this study. The study was done, the release, we actually did it up in Geneva at the experiment station. And it was the release of the virus into the cabbage field, which were seeded with cabbage loopers, was done by Tony Shelton. Tony had a license to apply pesticides. I had no such license. The other part of the study was done with Pat Hughes, who was also a member of the Boyce Thompson Institute. Pat probably had the highest IQ of any scientist I knew at Boyce Thompson. He was really important to our work. So in 89, we did the first field release. The next day, it was all forgotten. I mean, talk about quiet. I mean, the phone stopped ringing. It was. Oh, I should also mention the movie or the television things. We were on the Today Show for 5 minutes. NBC's Today Show. Yeah, the board at the institute was ecstatic. They'd never had that kind of exposure before. But anyway, so it went dead silent. Nobody. So I go back to EPA and say, okay, they only could give me money for 1 year. I need the second year in order to go out and evaluate the field. How well did the experiment work? They wouldn't give me the money. Ralph Hardy was the director of the institute at that time. Ralph said, it's important, I'll pay for it, you do it. So Pat and I went out and we evaluated the next year how things were working. The experiment failed. It didn't work the way we thought it would. It didn't cause any problems, but so we sat down and started thinking about it and said, well, there's another way of doing this, a better way. The experiment failed, but that gave us the information we needed to make a better thing. And from that came the way to make recombinant proteins in insects. And we formed Agrovirion. It was the first spin-off, as far as I know, the only spin-off company ever out of Boyce Thompson. A few years later, we sold it, the company to Chesapeake Pearl, and we're scientists. We're doing research. This is not our thing. We're not businessmen. So we make recombinant proteins in insects, and it's much cheaper to produce them in insects than it was in bacteria cells or in yeast. And we became very interested is that if we were going to make them in insect cells, maybe they could be properly processed. Bacteria cells cannot produce glycoproteins. They cannot put sugars on proteins. Yeast can put proteins on, but they put on this ridiculous thing that we rejected by a human body. So we got interested in what would be the value of having a recombinant protein produced in an insect cell. Maybe it could be glycosylated properly. So we got a whole lot of cells and started looking at them. That's when we got cells from Bob Granados and found that the high 5 cells were just huge amounts of protein. Gave it back to him. We're interested in glycosylation, and we're going on. It turned out that under conditions of microgravity, which we were able to, working with NASA now, we could have significant effects on the glycosylation, and in fact produce glycoproteins that had what they call a lot of terminal sialic acids, which is a very important terminal sugar in human cells. Like 90% of the proteins in your blood system are glycoproteins. So I go to NASA and I go in and I explain to him what we had found in glycosylation with these insect cells and what the potential effect was on it. And the project officer says he knows about glycosylation. Glycosylation is not for the weak at heart. I mean, this is not a simple subject at all. I mean, I had to go back and do a lot of organic chemistry in order to get back into this. So he knows about it, and he said, "Well, we can go into space and we can produce new proteins now in space, maybe." I said, "No, that's not why we did this. We went to it because we know that astronauts are being affected by being under microgravity. When they go up just for a few hours, they come back anemic. The reason they're anemic is because the erythropoietin that's produced in the liver, where is it produced? I've forgotten now where it's produced. But anyway, it's produced, it circulates. If it's not glycosylated properly, it will be sequestered by the liver. If it is glycosylated, it goes to the bone marrow and you make red blood cells. If it's sucked off by so I said, give me a few cells from an astronaut. The technology we got it from England. Just need a few cells to do this. We'll look at the surface proteins on those before, during, or after the flight. I know you've got tons of them. No. You need to go. We need to think about going into producing proteins in space. Our grant was not renewed. Well, no, it was a fun project. It was real interesting. Yeah. That kind of came to the end of Boyce-Thompson, really.
How did the patent you filed affect insect virus production?
The patent was really important to production of proteins in insects, because in the past, what people had to do is they would take each individual insect and they would inject it with virus in order to infect them, one at a time. The patent that I put in was included in a technology where you could just contaminate the food, they would feed on it and get infected. It was actually a new form of the virus that had never been recognized before. It's called pre-occluded virus. And in fact, I don't even see it in the literature. Nobody ever picked up on and uses this. It turns out that most of the virus never, well, I don't know if you're familiar with insect viruses, they get occluded in these crystals. And that's the form of the crystals that are fed to the insects. And there's also a form of the virus that gets excluded, buds out of the cells. That form of the virus won't infect an insect unless you inject it in, and it will infect insect tissue culture cells. There's about 90% of we found, anyway, with Pat Hughes doing this, we found that about 90% of the virus that's produced never becomes occluded and never gets budded. It's just inside the cell. And it's the form of the virus that if it was fed orally, it would infect them. Bingo. So it became much easier. I mean, that's what Chesapeake Pearl wanted there. It's like, yo, you got a patent on this. This is the way to do business.
What's another notable time at BTI you feel proud about?
So we can produce recombinant proteins in the insects themselves. And so we got together with the people over at the vet school to look at, could we insert the genes for the coat protein of the canine parvovirus and express it in insect cells, and then use that as a way of vaccinating dogs against the canine parvovirus. It worked. In fact, it was the only parvovirus that had been successful at that time. We were at a meeting one time having dinner, and they said, "Oh yeah, well everybody's tried to do that. You were able to be successful with a parvovirus?"
How did an interaction with one of your technicians impact your life?
One of my technicians joined the suicide prevention organization in town, and she convinced me to give it a try, which I did. And I ended up, actually two interesting things. One, I ended up on the board, and second of all, I chaired the group that was able to buy the building that they're in. But I was training somebody one night. You get on the phone and you listen in while the trainee is going through. It was like 40 hours of training before you can get on a phone, at least in those days. And during this discussions in between phone calls, this woman said, "Oh, do you date?" Well, I'd been divorced a few years before that. And I said, "Yeah, I date." But and she said, "Well, I know this woman, maybe, fine." And, I asked her if she'd like to go out. I ended up marrying her.
What made BTI's greenhouses special during your time here?
In those days, it was really strange. For recombinant plants, genetically engineered plants, the only place on campus that was that we would allow those plants were at Boyce-Thompson. Our greenhouses were the only one that met the criteria. Now you can grow them anywhere. You can grow them in your backyard. Nobody cares. They're really very cautious.
Any final thoughts you'd like to share?
One last thing.
Yes.
If you have to go to work, find something else to do.
That's great.
That was always my philosophy.
Yeah.
I wasn't going to work. Yeah, I was really lucky.
Yeah.
I look back and I was at the right place. I mean, when I joined the Institute, I was going to be here for 3 years. That was it. I did not join their retirement program. I was going to be history. I was going someplace. They didn't want me to write grants. In those days, when I joined, they still write grants. So I'd write grants because I want to be able to get another job someplace. And I ended up, and didn't leave, obviously. But yeah, that was not the original idea.